Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. So in this video tutorial, as we have already discussed, that we will going to do a lecture on N-terminal analysis of a peptide, right? So N-terminal, first we have to learn what is the N-terminal, what is the C-terminal. So as let's consider a dipeptide consisting of two amino acids. Okay, so these two amino acids are joined together via one peptide bond, right? So now the carboxyl group, which is not involved in any kind of peptide bond formation, is present in the C terminal in, okay, and free amino groups, or the direction where free amino group is present, we will call it as N terminal. Okay, that means the direction of the free amino group of the amino acid which is not involving in any kind of peptide bond formation will be known as N terminus, and the direction where free COH group is present we will call it C terminus. Okay, so now we will going to understand the N terminal analysis of the peptide. So two kind of reagent are available to find out the N terminal analysis of amino acids or N terminal analysis of a long peptide or protein. Two type of reagents are available for determining the N terminal analysis. One is 1-fluoro-2,4-dinitrobenzene, commonly also known as FDNB. Another one is dancyl chloride. Both are performing the thing in the same manner. So if we understand one, then we can already guess the second one. So let's move. So FDNB or 1-fluoro-2,4-dinitrobenzene works in alkaline solution of pH 9.5, and it usually works with the free amino group of the N-terminal amino acid residue, because I have just told you that N-terminal is the direction of the free amino acid residue where there is where the amino group is not involving in any kind of peptide bond formation, right? So this free amino group will going to react with this FDNB, okay, in alkaline solution, and there will be removal of HF, okay, that ultimately gives us a yellow color compound, dinitrophenyl derivative. So once we have derived the yellow dinitrophenyl derivative, now it is important to remove that FDNB compound from that peptide, and we can easily done this with the help of acid hydrolysis or enzymatic hydrolysis. Okay, then by subsequent analysis we can easily find out what is the amino acid composition of the N-terminal amino acid residue. Okay, so scientist Sanger first used this method to determine the primary structure of the polypeptide hormone insulin. That is why this reagent, that is the 1-fluoro-2,4-dinitrobenzene, is also known as Sanger's reagent. Okay, so another important thing, that is our dancyl chloride, will going to do the same kind of thing. It will also going to react with the free amino group of the N-terminal amino acid residue and produce a strongly fluorescent derivative of free amino acid or an N-terminal amino acid residue of protein. That means it will going to form a complex that have the fluorescent properties, and then by fluorescent measurement techniques we can find out which amino acid it is, right? So if you see this picture, this is a dipeptide of glycine and leucine. So FDNB will going to react with this free NH2 at the N terminal of the peptide, and there will be removal of HF, right, as you can see. This F will going to take one hydrogen from here and liberate it in the form of HF, and there will be a bond formed, right? So this is the complex that we are calling as yellow dinitrophenyl derivative, right? Now then acid hydrolysis, we can remove the FDNB containing N-terminal amino acid. So that is the idea of how the thing can be done. Okay, so now this is the densyl chloride. So here I have taken example of amine because we just only need the NH, so we can also take this example. In case of NH2, the example is same, right? So this is the structure of densyl chloride. You have to remember that ultimately gives us a strongly fluorescent derivative, okay, that we can measure with the help of any kind of fluorescent techniques. Okay, so that is the idea. So that is the video for our N-terminal amino acid detection of peptides. Okay, so that is the thing. So for a quick recap, 1,fluoro-2,4 dinitrobenzene will going to react with the free amino group of the N-terminal amino acid residue of the peptide and gives us a yellow colored dinitrophenyl derivative. Now this yellow colored dinitrophenyl derivative can be removed by acid hydrolysis or enzymatic hydrolysis from the rest of the peptide bond, and then by further analysis we can find out which kind of amino acid it is, right? Sanger first used this technique to determine the structure of the insulin. That is why this reagent is known as what? This reagent is known as your Sanger's reagent. And densyl chloride also do the same thing, but it will use fluorescent properties, right? So that is the idea of our N-terminal analysis of peptides. Hope this video will be helpful for you, and more importantly, you have to mention the alkaline solution. Okay, it is necessary condition. So that is the idea. So I hope this video will be helpful for you. Thank you.